Now today, the topic for discussion is the lung surfactant system and its behavior in health and disease. What I'll try to do is to cover some history and then bring the subject up to date, I hope. If we can begin with the first slide. I'd like to acknowledge the Swiss physician Kurt von Neergaard, who first demonstrated the importance of surface effects in the lungs in 1929, and who also hinted that the alveoli might contain surface active material. Somehow his papers were neglected until 1953, when Ted Radford and his colleagues at the Harvard School of Public Health became interested in lung surfaces and later found von Neergaard's writings. But Kurt von Neergaard had died in 1947 without seeing any application of his discoveries in medical practice. You might well ask, why should that be? As a young man, he was an ardent researcher. Though trained as an internist, he spent most of his time with his friends in the physiology department at Zurich, investigating physical factors in lung function. His findings had basic importance, but his medical colleagues ignored them. Disenchanted, he withdrew to the Institute for Physical Therapy in Zurich and contented himself with speculations on the interrelationships of philosophy, politics, medicine, and social welfare. Today, about 40 years after his death, the world of medicine can no longer ignore him. The application of his discoveries to clinical problems has now become obvious. Prominent among these is hyaline membrane disease. This disease, also called respiratory distress of the newborn, is characterized by progressive lung collapse and pulmonary failure. And as Dr. Massaro has mentioned, it's the principal cause of death in the newborn. The worldwide incidence of this disease is still estimated at about 250,000 cases a year. But I think it is true that the mortality has dropped 10 to 20-fold in the last 15 to 20 years. Well, now, during the rest of the talk, I'd like to tell in part what Kurt von Neergaard's discovery was and how his legacy has been amplified, and especially how work done by my colleagues and myself have developed that further. So let's start first with the question, what was von Neergaard's insight? He realized that the lungs are convoluted into very tiny air spaces whose cumulative surface area is huge and whose surfaces are wet. It's as if the tracheobronchial tree arborizes into millions of miniature bubble pipes, each with an alveolar bubble blown on its bowl. And he recognized that the surface tension of such structures should tend to make them decrease in area and should urge them towards collapse. He reasoned that the strength of this tendency could be estimated from the difference in the distensibility of the lungs with the interface between the parenchyma and air present and with it absent. To remove the interface, he filled excised lungs with isotonic salt solution so that the moist lungs could be filled with a watery solution. No, that's forward here, okay? There we go, thank you. Now this illustration shows how his experiment turned out. It's a graph of the volume of the lungs plotted against the pressure they exert. As you can see, the pressure was much higher at any volume when the lungs were air-filled, here, this curve, than when they were filled with isotonic salt solution. In other words, when the alveolar surface tension was adding to the retractive force of the lungs. This result has been confirmed many times, and in fact, the data you're seeing here were gathered by Radford at the Harvard School of Public Health using the lungs of a cat. One finds the same kind of results with the lungs of any mammal, including man. Radford, a physiologist who was first trained as an engineer, analyzed this pressure volume diagram in greater detail. He knew that the area between the return curves, here and here, represented the elastic energy stored in the lung, and therefore the area between the two curves equaled the elastic energy stored in the alveolar surfaces. He also knew that that energy was the product of the area and the surface tension. So if he could estimate alveolar surface tension, he could calculate alveolar surface area, and that was his interest, his main interest in doing this experiment. To do that, he assumed that the moist surface of the alveoli was composed of extracellular fluid with a surface tension of 50 millinewtons per meter, dynes per centimeter, as we used to, units we used to use. He was astonished to find that the area so calculated was only 1/5 to 1/10 of prevailing histologic estimates, and he concluded that the histologic estimates were in error. When I encountered Radford's work, I was delighted by his application of physics to this problem of lung function, and so I studied his paper thoroughly to learn whatever lessons I could from it. As I played with his analysis, I got increasingly unhappy with his conclusions and finally decided that they really couldn't be correct. It seemed unlikely to me that the histologic estimates of lung area really could be 5 to 10 times too high, and also his estimate of area seemed too low to me to accord with known diffusion capacity of the lung for gases. So when I applied Laplace's tension, radius, area, pressure formula to these data, using what I knew were accurate estimates of alveolar size and pressure, it was my turn to be astonished. The calculated surface tension fell to very low values, as you can see in this plot of surface tension against the percent volume of the lungs. And those values fell far below the 50 millinewton per meter value characteristic of extracellular fluids. Well, if alveolar surface tension really did approach zero, the lung must contain a wonderful kind of soap to achieve such a result, and perhaps it could be extracted. By that time, I had read enough about the physical chemistry of surfaces to know that I needed a surface balance to demonstrate the kind of a surfactant which changes surface tension as the size of the surface varies. I didn't have a fancy surface balance of the kind physical chemists use, but I was able to knock together a crude one out of sealing wax and string and the sorts of things that abound in old-fashioned physiology laboratories. And it worked well enough to show that extracts of normal lungs, but not of other organs, do contain a powerful surfactant, which causes surface tension to vary as you change the surface area in the surface balance. And in fact, to make the surface tension approach zero, as that graph in the lower part of the slide suggests. While these studies were going on, reports came in that Richard Pattle, a British toxicologist who had originally been trained as a physicist, had postulated the existence of a strong surfactant in the lungs. He had deduced this from the extraordinary stability of the bubbles in the foam, which wells up in the airway in animals suffering pulmonary edema due to inhalation of phosgene. I was somewhat dubious about his methods, but his results pointed the same way as our results, and so they were a powerful stimulus to continue. So my colleagues and I began to mull over some obvious questions about such a surfactant. What is its chemical nature? Where does it originate in the lungs? What molecular properties account for its extraordinary surface activity? How is surface tension regulated in normal lungs? Could deficiency of the surfactant explain some syndromes characterized by lung collapse? Are any diseases associated with an excess of lung surfactant? We debated these questions, did additional experiments, developed a theory of alveolar mechanics, and presented our results at meetings of the American Physiological Society for the next 3 years without receiving the impression that anyone was listening seriously. And we began to wonder if the scientific world was going to ignore us as it had von Neergaard. When I submitted my paper to Science, it was rejected. Dear contributor, we're returning your manuscript. It does not suit our present needs. P.S. We note that you sent your story by first-class mail. Junk mail may be sent third class. Now, one's feelings at such a time are a complex mixture of frustration, superiority, self-doubt, contempt, and a desire for a spectacular immolation. Fortunately, somebody was listening. Jerry Mead, a physiologist previously trained as an internist, heard and understood the significance of our experiments. And when Avery, a pediatrician especially interested in respiratory diseases of the newborn, went to Harvard for a fellowship in physiology, Mead sent her to my laboratory to learn the methods that I had developed. Well, to her credit, Dr. Avery was properly skeptical of my homemade surface balance, but she took the methods back to Mead's laboratory, and they made a better surface balance. And they applied the methods to the lungs of infants obtained at autopsy. Soon they published a paper reporting 2 important findings. One, that surface active material was not usually demonstrable in normal lungs, that is to say, lungs of infants that had not died with respiratory disease, whereas in those that died with hyaline membrane disease, the surface tension was not low, regardless of their gestational age, their birth weight. With this demonstration of relevance, the world of medical science began to take notice, and other investigators began to enter the field. We became very curious about what molecules make up lung surfactant. Many investigators have worked on the question, but I will limit myself mostly to what has been done about it in our laboratory in San Francisco. Our first step was preparation of surfactant from freshly excised cow lungs by pouring isotonic saline down the airway and breathing the lungs until copious amounts of froth issued from the trachea. This was a messy experiment that required a big sink and hip boots, but it gave large amounts of strongly surface active, albeit crude material. Klaus, Havel, and I were able to collect the foam and wash it with distilled water, dry it down to a fluffy, somewhat waxy white substance with a fishy odor that retained powerful surface activity. Some people still think it's fishy. The waxiness suggested that the substance might contain lipids, and indeed we found that much of the material was soluble in chloroform. In rough experiments with crude extracts, I had previously found that incubation with trypsin destroyed the surface activity, so we surmised that the active substance might be lipoprotein in character. Thin evidence, but I guess, as it turned out, good enough. However, the delipidated protein moiety was not active. Using a combination of chromatographic and analytical techniques, we found common kinds of lipid in our material: phospholipids, cholesterol, free fatty acids, triglycerides. The surface active fraction was largely phosphatidylcholine, and this was uncommonly rich in palmitic acid, a saturated fatty acid containing 16 carbon atoms. And the phosphatidylcholine fraction was strangely poor in unsaturated fatty acids. Surface activity, which disappeared in the intermediate steps of fractionation, reappeared in the phosphatidylcholine fraction. And then, fortunately, although I admit it seemed unfortunate at first, our preparations of surface active material varied in potency from time to time. And finally, it became clear to us that when the preparations were better, the phosphatidylcholine fraction contained more palmitic acid, sometimes as much as 75%. To explain this more clearly, let me remind you of the structure of phosphatidylcholine. It consists of glycerol, esterified on the first carbon with phosphorylcholine, on the second carbon with a fatty acid, and on the third carbon with a fatty acid. And as phosphatidylcholine normally appears in tissues, this fatty acid esterified at the 2 position is usually an unsaturated fatty acid, and the fatty acid at the 3 position is commonly saturated. And as you obtain mixtures of phosphatidylcholine from natural sources, they are combinations of many different species of phosphatidylcholines. You can see, though, that if palmitic acid makes up 75% of the fatty acid residues in the phosphatidylcholine, then at least half of the molecules must have palmitic acid in both the first and second positions. In other words, at least half of the fraction would consist of a single molecular species, dipalmityl phosphatidylcholine, or as we tend to abbreviate it, DPPC. And this is the molecule depicted in this illustration using CPK space-filling atoms. The prominence of this unusual phosphatidylcholine in the surface-active material led naturally to the hypothesis that it was the core of the surfactant, and this hypothesis is generally accepted now. Luckily, synthetic DPPC was available, and it was easy to show that it is indeed a powerful substance in lowering surface tension. By itself, DPPC absorbs from the water phase to the interface too slowly to be an effective lung surfactant. Several years ago, Alec Bangham, a biophysicist originally trained as a pathologist, was visiting our laboratory. He mentioned that in his experiments, ordinary phosphatidylcholine formed films quite slowly, so we decided to try DPPC. We put an emulsion of DPPC in the homemade surface balance and set it to working. Despite a 100-fold excess of DPPC in the water, a film had still not formed after 24 hours. That seemed too slow to be of much use to a newborn infant taking his first breath. To prove that DPPC was still around, we passed alcohol vapor over the surface, and in about 20 minutes, a good film had formed. The surface tension dropped almost to 0, and it continued there when the alcohol vapor was removed. The experiment brought out 2 interesting things. First, the natural pulmonary surfactant is not likely to be pure DPPC because DPPC forms films too slowly. And secondly, DPPC films can be stable in the presence of ethyl alcohol, a fact that became useful later. It's always interested me to speculate on what Senator Proxmire might have said if he'd come into the laboratory at that moment and seen a rather peculiar English gentleman and a rather immature and silly American scientist playing around with a drunken film of DPPC in a homemade surface balance. I think he might very well have awarded his Golden Fleece award, or he might have asked what has that to do with lung disease. Probably if that had happened, Alex Bangham would have answered that A real scientist works on what interests him, not on what interests other people. Anyhow, at that juncture, it became obvious that we needed more gentle and specific methods for isolating and characterizing the natural surfactant. It seemed likely that the surfactant might be as complex as tissue and serum lipoproteins were already known to be. King and I developed a method based on density gradient centrifugation, which yielded a lipoprotein having almost theoretical surface activity. The physical properties and the chemical composition of this material were physiologically acceptable and were also reproducible from preparation to preparation. Analysis of the phospholipid and neutral lipid components showed them to be similar to those of membrane lipoproteins, except, of course, for the large proportion of DPPC and phosphatidylglycerol, another lipid that's unusual in mammalian tissues. We wondered why lung surfactants should have such a complex composition, and we still wonder about that. John Girk, Jim Hildebrand, and I investigated the relationships between the various phospholipid and neutral lipid moieties and the physicochemical behavior of films formed by them at the air-water interface. It's now clear that DPPC alone excuse me, cannot perform all the necessary functions. Other components are essential, especially for the rapid formation of surface films when new interface is created by expansion. Phosphatidylglycerol has been thought essential for activity of lung surfactant because its appearance in the human fetal surfactant correlates well with maturation of surfactant function, as measured by low risk of respiratory distress at birth. Beppu, Girk, and I fed rabbits myoinositol, which in sufficient amounts suppresses phosphatidylglycerol synthesis and enhances phosphatidylinositol formation. The animals had normal pulmonary function and their lung surfactant was normal in all respects, except that it contained phosphatidylinositol in place of phosphatidylglycerol. And we, at this juncture, have still not found a function that seems unique to the phosphatidylglycerol component. On the other hand, the apoproteins, represented by this 8% of how your taxes are spent, have proved especially intriguing. Using the density gradient procedure, Dan Klass prepared surfactant and raised antibodies against it, and he showed the antigens were lung-specific and were localized to alveolar spaces. Richard King proved that the apoproteins contained the antigenic sites to these for these antibodies, and he characterized the proteins by extracting them from surface active material isolated by the density gradient procedure. He paid most attention to what he called the 34,000 Dalton, the 34 kilodalton group of proteins, but he also demonstrated a relatively low molecular weight group of proteins, which he referred to as the 11 kilodalton group. We now refer to these as SP 2636, 26,000, 36,000 group, and SP 5 to 18 group. King developed sensitive immunoassays for it, and then when these techniques were refined further, Mary Williams and Brad Benson in our group showed highly specific localization of lung surfactant antigen by electron microscopic immunocytochemical methods. In the endoplasmic reticulum, the Golgi apparatus, multivesicular bodies, lamellar bodies of alveolar epithelial type 2 cells, in alveolar spaces, and in phagosomes of alveolar macrophages. Sueishi, Benson, Williams, and Girk found that calcium ions and this SP 2636 group were absolutely required for the formation of tubular myelin, a peculiar lung surfactant structure, which I'll speak about a little more in a moment, and for the fast generation of surface films by lung surfactant components. In the last few years, Hallgood and Benson and more recently others in other laboratories have purified the 34 and 11 kilodalton protein groups to homogeneity. And using materials from our laboratory, scientists at California Biotechnology Incorporated have determined the complete amino acid sequences for these proteins. This is the sequence for the 2636 kilodalton group. The human gene for this group has been cloned when introduced into Chinese hamster ovary fibroblasts. It's able to direct the synthesis of the SP2636 group of proteins, and these, when isolated from the culture medium, work properly in vitro. This is the sequence as presently known for the main protein, the so-called SP18 of the SP5 to 18 group of low molecular weight hydrophobic surfactant proteins. This has also been purified, sequenced, and cloned. The gene product that is depicted here has a molecular weight of approximately 40 kilodaltons, so clearly it's a precursor protein. And the nature of the post-translational changes, which generate the low molecular weight 5 to 18 kilodalton hydrophobic proteins, have yet to be worked out. Well, why should there be two kinds of surfactant apoproteins? I suppose it'll be a long time before we can answer that question fully. However, Sam Hallgood has already shown a fascinating synergy between the two groups of proteins. With either protein alone added to surfactant lipids, this is making reconstituted lipids as liposomes and putting them in a surface balance to see how quickly a film forms. That's indicated in the vertical axis with respect to time. The lipids by themselves are very slow. When the SP2636 group is added, the speed is increased somewhat. When the low molecular weight hydrophobic protein is added, it's increased more, but film formation becomes virtually instantaneous when both proteins are added together to the surfactant lipid recombinants. We think, and many others believe too, that this is a critical characteristic of a surfactant. When the newborn baby takes its first breaths, there's only a fraction of a second for the surface film to form. If the surfactant is too slow, the film doesn't form well, and on exhalation, the lung collapses. Every breath becomes a first breath in that sense, and the lung tissue becomes severely traumatized, leading to hyaline membrane disease. We're now testing artificial surfactants made with these two proteins in fetal rabbits delivered before their lungs contain surfactant to see if the synergy works in vivo. I don't yet have the final results, but the initial results are very encouraging. Well, now returning to endogenous lung surfactant again, let's consider the question, where is lung surfactant made? As long ago as 1954, Charles Macklin of the University of Western Ontario deduced that certain large granular cells which occupy the corners of the alveoli secreted material in the alveoli, in his words, to regulate their surface tension. We see these cells here in a retouched figure due to Sergei Sorokin, made obvious by staining for the enzyme glucose 6-phosphate dehydrogenase, an enzyme which they possess in abundance. We now tend to call them type 2 alveolar epithelial cells. And here, seen in an electron micrograph of Mary Williams, is a type 2 cell. You can see that the granules are really osmophilic inclusion bodies or lamellar bodies. You can see their lamellar appearance and higher magnification, and that suggests to electron microscopists that they are rich in phospholipid. The contents of the lamellar bodies are secreted by exocytosis into the alveolar lumen. Here you see a stoma formed at the apical surface of the cell, and here you see a more advanced stage of exocytosis with lamellar body contents actually leaving the cell into the alveolar fluid. Now, once out of the cell, they form a structure which may not show too well in this picture, called tubular myelin. And that's a fascinating and beautiful kind of structure. Here you see it at higher magnification. The phospholipid bilayers intersect at right angles. So far as I know, this is the only structure in nature that in which membranes do intersect at 45 degrees. I think if you look closely, you may be able to see particles that occupy the corners, which we believe represent the larger molecular weight apoproteins. And we think that the small molecular weight hydrophobic apoproteins occupy the voids in the intersections where the bilayers meet. Anyhow, that's a very speculative description of the structure of tubular myelin. It's weird and wonderful, and will bear a lot more study. Type 2 cells have been isolated and cultured by several groups. Mason and Dobbs in our laboratories found that beta-2 adrenergic agents, calcium ionophore, protein kinase C activators, and phosphodiesterase inhibitors all stimulate these cells to secrete surfactant components, whereas cholinergic agents and prostanoids are ineffective agonists for secretion. Dobbs and Benson also showed that the phospholipids secreted by these cells in culture are virtually identical to those found in lung surfactant isolated by lung lavage. And that gives us some comfort, since we always worry that material washed out of the lungs is contaminated with materials from sources other than lamellar bodies. The isolated cells also synthesize, have been shown to synthesize the sp2636 group, and I think Whitsett and his group have shown that by in situ hybridization techniques, that message for at least one of the low molecular weight hydrophobic proteins can be found in type 2 cells in intact lung. Now, beta-2 adrenergic agents also stimulate secretion of surfactant in vivo, and it's especially interesting that deep breaths can cause surfactant release, and that beta blockers inhibit that response. Since breathing uses up surfactant, this phenomenon probably signifies the existence of important feedback regulation of the amount of functional surfactant in the alveoli. Well, it didn't drop. I want to show you the results of experiments that Lee Dobbs and his co-workers have done using cultured alveolar epithelial type 2 cells and stimulating them to secrete surfactant with TPA, tetradecanol forbyl acetate, or with beta-2 agonists. Have we lost the bulb? As the slide is supposed to show, applying increasing amounts of surfactant apoprotein SP2636 to the cells in the culture medium progressively inhibits secretion. That's the slide. Here we see increasing amounts of surfactant protein on the x-axis and the percentage of stimulated secretion, stimulated by, well, PMA, phorbol myristate acetate here, so that it suggests that the major higher molecular weight surfactant apoprotein can inhibit surfactant secretion by type 2 cells. Now, of course, this is an in vitro system. We don't know whether that same effect occurs to a significant extent in the lungs. But nonetheless, it's interesting and certainly is a very important finding to those of us who think about what the composition of artificial surfactants should be for surfactant therapy. In any case, the turnover of lung surfactant, it worked, is normally very fast, as you can see by injecting radioactive precursors to surfactant components intravenously. The label appears very rapidly in lamellar bodies and somewhat later in alveolar surfactant. And then the specific activity declines relatively slowly. The labeled surfactant stays around a long time. Fisher and Bartusio and I found that the amount of labeled lung surfactant secreted into alveoli far exceeds that taken up by alveolar macrophages or appearing in airways. And so that led to the question, where does the material go? Clearly there's a turnover, a secretion of material, which is quite rapid, but it's not leaving to a significant extent through alveolar macrophages or up the airways. Electron micrographs have not been of any help in this regard. They don't really show any obvious pathway for the exit of the surfactant from the alveolar region. The first clear demonstration that surfactant components might go back into type 2 cells, the cells that had secreted them, came from autoradiographic studies. Klaus Geiger at Harvard gave radioactive DPPC by aerosol, by inhalation, to rats, and found that the label moved quickly from the alveolar lumen into type 2 cells. Then Manuel Oyarzun and I showed that radioactive DPPC, which we instilled into the lung via the airways, was taken up by the tissue with a half-time of 3 to 4 hours. We found out that the uptake was not stereospecific. In other words, the DPPC molecules were probably absorbed intact without the necessity for enzymic degradation. Double label studies have confirmed this conclusion. Most of the label, however, stays in the lung in DPPC for a relatively long time. So the question is how? Mary Williams has recently demonstrated an alpha-galactose inhibitable vesicular uptake pathway, which leads from the alveolar lumen into the lamellar bodies of the type 2 cells. Now, whether surfactant components travel this same pathway still remains to be proven. What we do know is that when SP2636 group proteins are added to liposomes of surfactant-like lipids, the uptake into lamellar bodies, in intact lungs, and into type 2 cells isolated in vitro is greatly enhanced. Here we see the uptake of surfactant-like lipids, and here when the higher molecular weight surfactant protein is added to them. So it looks as though SP2636 also enhances uptake of, could enhance uptake of surfactant materials from the alveolar spaces into type 2 cells. One can calculate from the tracer data, which I showed a couple of slides back, that more than 85% of the secreted phospholipid components can be recycled into lamellar bodies. Following clues provided by Don and Gloria Massaro, we've recently subfractionated alveolar surfactant into several forms that have comparable phospholipid composition, but different protein components and very different surface activity. And currently we're investigating the relationships between these forms and the recycling of lung surfactant components. An important implication of these results is that in normal circumstances only part of the phospholipid in the alveoli is surface active, and perhaps no more than 30% at some times. Clearly, we need to learn a lot more about the mechanisms, pathways, and control of lung surfactant recycling and clearance, and about how lung diseases affect these processes. This slide is a cartoon which summarizes how we think about lung surfactant metabolism these days. The alveolar epithelial type 2 cell, the type 1 cell, where is it, over here, and an alveolar macrophage. Here's an airspace up here, a capillary, pulmonary capillary lumen here, interstitium here. Precursors for surfactant formation presumably enter from the circulation and are incorporated into the phospholipid and protein components in endoplasmic reticulum, packaged in Golgi and multivesicular bodies, and stored in lamellar bodies, following which they can be secreted by exocytosis into the liquid that lies on the epithelial surface, where we postulate tubular myelin forms are generated, from which very rapidly, when the lung is expanded and an interface is generated, a monomolecular surface film can be formed to lower the surface tension and stabilize the air spaces on expiration. What happens to the material after it has performed this stabilizing function is not really known. Some freeze fracture images suggest that small vesicles may be formed from the surface film. Certainly there's data that indicates that alveolar macrophages can take up some material, both lipid and protein components of surfactant. But in normal steady state circumstances, it would appear that most, at least of the phospholipid components, re-enter the type 2 cells, are repackaged. And this is the vesicular pathway which Mary Williams demonstrated using ferritin-labeled lectins. So we think that most of the material is recycling in this way, a small fraction, but nonetheless, over the long term, a very important fraction may be removed by alveolar macrophages, or possibly through other routes as yet not demonstrated. Well, while the lore of lung surfactant was building, a great deal was being discovered about lung development, about the appearance of lung surfactant in the fetal lung, and about respiratory distress syndrome. Mount Liggins in New Zealand set the stage for study of some of these subjects by working out elegant techniques for chronic in utero measurements on lambs and for observing the effects of hormones on lung development. Using his methods, we found that the output of lung surfactant in lung fluid of fetal lambs began at about 130 days of gestation and greatly accelerated thereafter. Well, we also found that administration of dexamethasone would cause precocious onset of lung surfactant release in fetal lambs. Following up on Liggins' observations, several research groups have elucidated cellular mechanisms of lung response to glucocorticoids, all the way from steroid binding on receptors to formation of the gene products. And these findings resulted in the successful use of corticosteroids in reducing the incidence of respiratory distress syndrome in infants born before 34 weeks of gestation. In this particular trial, a reduction in incidence from 28% to 14%. In conjunction with steroid prophylaxis, and in general, for the management of preterm labor, obstetricians need an assessment of fetal lung maturity, or conversely, of the risk of respiratory distress syndrome. Lou Gluck and his colleagues in San Diego showed that the surfactant components in amniotic fluid could be used to predict lung maturity in the prematurely born. And following his lead, we developed a simple, fast foam test for lung surfactants that's used in many hospitals around the world now as a screening test for respiratory distress syndrome. Here you can see that when there's surfactant present, you got a meniscus full of stable bubbles that indicate the presence of substantial amounts of surface active material in the amniotic fluid. And I would remind you that the design of that test, which includes the use of ethyl alcohol, was based on our earlier results with the effect of ethyl alcohol on DPPC. I don't think this might have happened if Senator Proxmire had actually visited the lab. Anyhow, we showed how gestational age and the results of the foam test together could predict the risk of respiratory distress syndrome. And in this slide, very crudely, the width of the black curves, shaded curves, indicates the uncertainty of the risk estimate when both estimated gestational age and foam test results are considered. For many years, pediatricians have been interested in the cardiopulmonary adaptations that occur at birth and in the derangements of these adaptations that accompany respiratory distress syndrome. Physiological studies of infants documenting the changes in the lungs and circulation that accompany birth and the development, as well as the resolution of respiratory distress syndrome, have led to concepts that relate lung ventilation, pulmonary blood flow, distribution of lung fluid, metabolism of lung cells, synthesis and secretion of surfactant, and pulmonary mechanical properties. Such interdependencies may work to induce, to exacerbate, or to resolve the structural and the functional abnormalities of respiratory distress syndrome. These concepts have forced many improvements in the management of babies with respiratory distress syndrome. Over the years, the physiological approach has gradually become transformed into intensive monitoring of blood gases and ventilation, blood pressure, and the many other variables that are now measured routinely in intensive care nurseries. Attempts have been made over the years to ameliorate the disease by administering surfactant or pulmonary vasodilators, but the first trials were only marginally successful. Vigorous resuscitation and positive end expiratory pressure as a method of splinting surfactant-deficient lungs are widely used now with impressive results. While intensive care, positive end expiratory pressure, and prophylaxis with glucocorticoids have combined to reduce the incidence and mortality of respiratory distress syndrome, many problems still remain. Steroids do not always prevent respiratory distress. Some infants that do get respiratory distress require hospitalization for prolonged intervals. And as many of you are well aware, there's a significant incidence of complications, such as air leaks, patent ductus arteriosus, intracranial hemorrhages, chronic lung disease, and behavioral and intellectual deficits in the survivors. So many investigators are continuing the quest for further improvements in therapy of respiratory distress syndrome. Carefully controlled trials of new treatments are underway, and evaluation of new surfactant substitutes have begun in several medical centers around the world. As indicated in this graph of arterial blood oxygenation, Fujiwara found that instilling bovine surfactant into the lungs can dramatically improve pulmonary function in the infant with respiratory distress syndrome. Similar results are obtained with a completely synthetic surfactant that we designed at the University of California in San Francisco. This latter substance is about to begin clinical trials in 10 hospitals in the United States and in some other hospitals in other countries. I think it remains to be seen how well surfactant substitution will perform overall as an adjunct to the resuscitation of the newborn, and whether it will bring a new set of clinical problems for neonatologists to solve. May we have the slide off now and the lights, please? It's fair to say, I think, that we've come a long way in the field of lung surfactant. What began as a physicochemical abstraction invented to explain the bizarre stress-strain behavior of the lungs has eventuated in the discovery of a whole new physiological system, the lung surfactant system. Without multidisciplinary research, the knowledge of this system could not have developed as it has. This development required ongoing, constant interaction between clinical and basic scientists. And this interaction just did not happen in the circumstances in which Kurt von Neergaard worked. Taking part in this research is a great adventure for me, and so I thank my lucky stars that I was not born 40 years too soon like Kurt von Neergaard, and I thank all of you for letting me share my enjoyment with you today.